So I'm going to begin with this question: how do you design nature? Which has proved an interesting question along the progress of this project, synthetic aesthetics, which I'm going to talk a bit more about in a while. But I'm going to talk about, I'm going to pose a set of questions about nature and how we understand the design of nature and our role in designing nature itself. As my background is in design, but although I call myself an artist, and that's something actually that I think that needs to be discussed as well. So this question, what's the difference between life and biology, is something that I think is also very important to open up in this discussion. As technology saturates our environment, it can be hard to distinguish nature from culture. The boundaries are long blurred. And now synthetic biology, which is this emerging field of genetic engineering, is applying engineering principles to the complexity of living systems and promising to literally yield the fruits of the promised biotech revolution. So one example of this is this plate of engineered bacteria. It's E. coli that's been engineered with the biobrick BBA K274,002 in 2009. So the 5 genes that make up this biobrick, which is a standardized format of for genetic engineering devised at MIT, are copied and pasted from the Chromobacterium violacum, which is a marine bacteria. And this was done by a group of undergraduate students from Cambridge University in 2009, 7 students trying it for the first time. Some of them never been in a lab before. And they devised a system of biobricks, which are these interchangeable parts of DNA that could be inserted into bacteria and produce different coloured pigments. And this project went on to win the International Genetically Engineered Machine Competition, which is a contest held at MIT every year where thousands of undergraduates get together to design bacteria. It's a flagship enterprise of the synthetic biology movement, sort of encouraging more and more students and more universities to get involved with this new brand of genetic engineering. Well, what for me is really strange about this project, and exciting as well, is that this week the project was nominated for Designs of the Year at the Design Museum, and I took part in this project with another designer, James King. We collaborated with the students, and now their engineered bacteria are sitting alongside iPads and light bulbs at a Fiat Cinquecento. And so nature has suddenly become a new site for design. It's suddenly been validated in a way that it hadn't before. So we're kind of used to this rhetoric of culture having been a subset of nature, and now the two worlds are becoming interchangeable. And Kirk van Mensvoort from Next Nature would argue that nature is even becoming a subset of culture. And it's clear, as Kevin Kelly said back in 1994, that machines are becoming biological and the biological is becoming engineered. We're used to the transition from the Industrial Revolution to this promised biotech revolution. But there's more to it than this, I think. We're no longer tinkering with biology, kind of swapping genes in and out. Synthetic biology is transforming not only biology, but life with it, into materials for design. And as a designer, that leaves me in an interesting place, and that's a lot of what my work has been about. I'm going to talk a bit more about the sort of ideology behind synthetic biology, because I think it's a kind of quite important topic at the moment. It's promising a shift from biology as a science towards biology as technology, as one commentator, Rob Carlson, describes it. But biology has up until now been descriptive, sort of the investigation and documentation and unraveling of the complex systems of life. Then synthetic biology is promising the construction of a design framework towards a prescriptive biology. So basically, in simple terms, it means that you can take DNA and abstract it and then standardize it so things can be pieced together. So instead of finding one gene from sort of a fish to put into a tomato and make a bespoke system, you can make a standardized toolkit. And if this is actually possible, the theory is that it would allow us to build biological machines for human needs more easily, rejecting the bespoke tinkering that's sort of seen in traditional genetic engineering. Abstraction, standardization, and decoupling from the molecular are its golden rules. Synthetic biology is rejecting the chaos and complexity of life in favor of rational engineering. We promise we can simplify the way we engineer life, making it predictable and easier to design. Parts and devices are interchangeable just like in electronic engineering, like the E. chromo parts I showed before by the Cambridge students. But this is all very well and good, and if these promises come true, we're going to have amazing impacts. So molecular scale designs happening at the scale of a microbe could have a global impact. We're already beginning to see processes like this emerging. So So the first new synthetic biotechnologies that are being developed include Jay Kiesling's biofuels. So a linear production system is being set up where bacteria are being engineered and they live in vats in California and they're fed sugarcane that's grown in Brazil. And then that's shipped up to America, the bacteria produce fuel or the yeast produce fuel, and then that's then distributed. But do we need to change our ways of thinking? We're replacing linear systems with more linear systems. We're actually engineering and designing into the molecular scale these kind of crazy systems where things are being shipped around and creating the same problems again. Do we need to think more, and sort of reflecting the earlier talk about thinking about ecosystems of design and new kinds of production that actually fit in with the design of nature? If we are designing it, these are living machines after all. And this quote, I think, is quite relevant here, and a lot of the debate around synthetic biology does actually sort of hinge on this argument between biology and engineering. And in fact, before the slide I introduced with the life versus biology, are they the same thing, or do we need to think of them in different ways? This was actually used when I first attended a synthetic biology course in Cambridge. This was the first slide that we were offered. Scientists discover the world that exists, and engineers create the world that never was. And Theodore von Karman was an engineer in the 20th century. This is another of my own projects, Growth Assembly, which was in collaboration with Sasha Poplak, where we were beginning to try and think about what this kind of growth in a multicellular plant, a multicellular organism could be like. When plants become machines, living objects become machines and are designed and engineered to produce things. This project describes 7 different parts that are grown as plants and then assembled together to produce a common product from this feature, which is a herbicide sprayer. And it's kind of introducing ideas of softness and diversity in a world that's dominated by manufacturing standards and industrial hardness. And one of the key points to this project is that you'd only have to ship seeds because they contain all the manufacturing instructions encoded into the DNA. So these kinds of questions are sort of framing this argument we're looking at in synthetic aesthetics, which is, what does it mean to design nature? Design has traditionally been in pursuit of production. Design has been acting as an interface or a moderator between a technology and the consumers it's created for. So how do you design nature? And how would you design nature? And one of the things that we're doing is asking, how do you design it well? Is that even a question that we can ask? So synthetic biologists sort of sit in this framework as engineers. Or do they sit somewhere in between the two? Or are they actually the new designers? Is my kind of training in design is it not going to be needed in this biotech revolution? Will scientists become the exclusive designers of living technology? Is this even design, or must we find new words to describe it? This image is another iGEM project from 2004, which is called E-Polaroid, and bacteria produced into a photographic film. So we might have to define new design tools to negotiate this changing interpretation of life and actually find new words for what this is. Is this new definition of science, this new definition of art and design? So the Synthetic Aesthetics Project, which I'll describe some of it now, was initiated by scientists, Drew MD at Stanford, Alistair Elphick at Edinburgh, and a social scientist, Jane Calvert at Edinburgh. And it happened in a sandpit for funding for new directions in synthetic biology. And it's really interesting that it was actually motivated by science. And the idea was to create a research project that can investigate what this new kind of design is and what it might hint at for future science, especially synthetic biology and culture. So bringing scientists, artists, and designers together, we're exploring this idea of shared territory, exploring, questioning, critiquing process, collaboration, and directions for future work. But what's been really important is that it's been set up as mutual exchanges. So for the talk earlier this morning was really kind of looking at this problematic relationship between art and science collaboration and how these two things can relate to each other and both find something useful has been absolutely key to this process. And I've been really lucky to be able to follow all these residents. We had 6 synthetic biologists and 6 artists and designers paired up, spending 2 weeks first in the lab and then 2 weeks in the artist or designer studio as their first kind of introduction. And the spread across this discourse has been incredible, from protocell chemistry to plant science, from bioarts. We had Oren Katz, Cecil Tolas, who's a smell artist, a critical designer, architecture. So I'll go through a few of the projects in the remaining few minutes. So we started off with David Benjamin, who's an architect in New York, working with Fernand Federici, who's in Jim Hasselhoff's lab at Cambridge University. And each of these collaborations started in a similar way. The artist or designer would come to the lab, first experience of the lab, learn the tools and techniques, and then the discussions would continue. What kind of shared territory do they have? What kinds of tools, processes, knowledge, and how do you start to develop a project in a synthetic way? We'd introduce these pairs together, and they were suddenly thrown into a foreign situation, having to find a way to make a project. And we hadn't actually set any boundaries for what kind of project it had to be. We told them they didn't have to make anything. It was more about the process of interaction. So Fernand and David started looking at these xylem cells, which are part of the plant growth system, plant water delivery system. And the cells grow into this structure, and then they die, and they leave behind this cellulose kind of structure where water passes through. And Fernand had become really interested in how do these cells compute? How do they decide what form to make? And they also discovered, Fernand and David, that they use a lot of similar tools. So Fernand was using 3D modeling and microscopy to create kind of 3D structures of the cells he's looking at. And meanwhile, this was back in New York at Columbia School of Architecture. David's students use exactly the same kinds of methods to kind of sort through large data sets and find patterns. So what they're looking at is how to kind of try and take the logic of this cell and the way it's built into architecture and into science. So they're using the idea of the logic of nature rather than the form of nature, so rather than biomimicry, to actually use the cell as a new kind of computer. So creating a challenge, so challenging definitions of synthetic biology as a metaphor for, as an analogous to electrical engineering. And they're taking it into architecture education. They're going to engineer cells and try and use them as kinds of computers. Now, the second collaboration is completely different. Christina is a PhD student at Harvard Medical School in the Silver Lab, and Cecil Tolas is a very well-known smell artist who's funded by International Flavors and Fragrances. And their project is now called Nothing Stinks, Only Thinking Makes It So. So Cecil's very interested in smell and how the world is becoming increasingly deodorized. And we only think that some smells are bad because we're taught to, whereas they're interesting. And there's actually some really interesting things about bacteria that I didn't know about, that certain cheeses have the same kinds of bacteria that your armpits or found in your feet. So they decided they wanted to start making cheese with human bacteria. So in the lab at Harvard Medical School, suddenly I was finding myself being swabbed, as was the social scientist following this project, and Cecil and Christina started making cheese out of all of our bacteria. We then also found, they were amazed to discover that there's a new lab being set up at Harvard where there's, the scientist is investigating cheese as a model organism. So the rind of cheese is specific, so like the ecosystem of cheese. So could it be used to be studied as ways of like microbial communities live together? So she actually had an amazing collection of cheese samples as well. So this kind of adventure continued to Berlin, where the samples had been shipped to Cecil's lab, because he's now back in the artist world. So this is Cecil's home. She's got this amazing collection of smell molecules. And the cheese kind of continued to develop. They made cheese from these different bacteria. We took him to a cheese shop, had an expert smell them. Their plan is then to start to engineer the bacteria in them, to really challenge this kind of boundary we have with the acceptable and the unacceptable. So this project is now, they're now looking for funding to continue this. So I think a few minutes left. Oren Katz, who's a bioartist, is working with Hideo Iwasaki, a Japanese scientist looking at cyanobacteria, and who's also an artist. And they're looking at stromatolites, which are natural formation in Australia. So the project is called the Biogenic Timestamp, and they want to engineer bacteria to kind of recreate these forms which are actually made out of cyanobacteria and form over many, many thousands of years. So they're looking at process of time and also kind of precise location of the geology of Western Australia and things like creating this whole kind of life cycle and ecosystem with engineered bacteria. We've got very quickly Will Carey, who's at IDEO. So this was a spread from the very kind of critical arts across to very product design, kind of commercial, was part of what we wanted to see, how the different things would work. working with Wendell Lim, who's the head of a big lab at UCSF. And they're looking at the process and how can you apply this IDEO design process to synthetic biology. And just a couple more slides. Sharif Mansi is a protocell scientist in Trento, working with Sasha Poflet, and they're really looking at the meaning of life, the shifting ground beneath the division of the natural and the artificial. Sharif is trying to create life from scratch. Part of that problem is when you know you've made it. So they need a philosopher in-house. So they've been looking at things like historic evolution experiments. So this is William Dallinger's controlled evolution experiment and other such experiments to start to look at the boundaries of a lifelike machine and how do we even begin to deal with creating and designing life in itself. And the last pair have just started, the musician and Chris Chaffee and Mariana Legueux, who's at Berkeley, and they're working together looking at large data sets in the recording studio, analysing data, and working with robotics, which is what Mariana does. She's high throughput sequencing. So I've kind of run out of time, but there's an interesting new sort of thing going on where we're looking at different kinds of relationships between science and engineering, art and design, and I think that the first talk this morning really kind of showed up the problems between these relationships. And I think what's happening in synthetic aesthetics is that there's something new that's being made that's neither science and it's not art or design, but it's somewhere in between the two. These projects have equal merit in both. They can both produce scientific papers and artistic projects. And that's something that's really exciting that's coming out of it. And we're hoping these projects will kind of challenge our understanding of synthetic biology, and as well as also challenging our definitions of art and design. I think that's a really important part of it. And ultimately a design to nature, whatever that is. Thank you. Sorry, I went a bit over time.